we're going to talk about our final topic, which is placental function. We're going to talk a little bit about the function of the placenta, especially with regard to hormonal function. We're going to talk about amniotic fluid, its purpose, where it comes from, and a concept of that involves basically things that can go wrong, namely being exposed to substances that can cause harm to the growing fetus, and that's a process that we call teratogenesis. So the placenta plays multiple roles for the baby. It plays a variety of roles, and we were talking about how in the last section how the placenta is really the lifeline for this growing fetus. So it has a variety of functions. It serves as a metabolic organ, so it contributes to the quality and the quantity of fetal nutrient supply. The placenta has a very high metabolic rate. It synthesizes things like glycogen, fatty acids, cholesterols, and enzymes that this growing fetus needs. It is a highly it plays immunologic function. It protects the fetus from pathogens. So especially in terms of the barrier between maternal blood and fetal blood, the placenta protects the baby from potential pathogens that may enter, including bacteria that are too large to cross the placental barrier but may be present in mom. It also prevents rejection of the fetus by mom. So towards the end of pregnancy, you may have some transfer of IgG or sort of the traditional antibodies, but really for the bulk of pregnancy, there's really no contact between maternal immune system and baby. But that transfer of IgGs, of antibodies, from mom to baby towards the end of pregnancy is actually designed for baby to have some passive immunity for the first few months of life after extrauterine life. And most notably, the placenta is an endocrine organ. It's really responsible for maintaining this pregnancy and inducing all of the metabolic adaptations and many other adaptations that mom makes in order to accommodate this growing fetus. So the placenta takes over the endocrine function of the corpus luteum when it's ready, and it produces a variety of hormones. And there's really four main hormones that the placenta is going to produce. It's going to produce HCG, or human chorionic gonadotropin. Okay? So human chorionic gonadotropin is a hormone that is produced very, very early on. You actually see it produced about 2 weeks after ovulation, and presumably also conception. And as you can see, once it's produced, the concentration of HCG doubles every day, and it hits a peak around 8 to 10 weeks of pregnancy. And then from there, it drops off dramatically. And as we spoke about in class, that rise in HCG is what maintains the corpus luteum, right? And its production of progesterone and estrogen until the placenta can take over. And you can kind of see around the same time when HCG drops, around the end of that first trimester, a little bit longer, you start to see the production, the placental production of progesterone and estrogen rise. Okay? So in addition to HCG, progesterone and estrogen are two major hormones that the placenta will produce. Progesterone is really going to be responsible for maintaining this pregnancy. And progesterone is so important for maintaining pregnancy that if you administer a a drug that blocks progesterone at any point during pregnancy, you will induce labor and uh, you will induce labor and end the pregnancy. And one thing that we will address as we later on is, if you notice, progesterone is usually higher than estrogen throughout pregnancy, except for the last few weeks of pregnancy. And this, when the tide turns, where estrogen levels start to exceed progesterone levels, you actually see this is actually a very long period of time, but it actually represents a period of time where the uterus is priming itself and getting ready for labor and delivery. And so estrogen is also an important hormone in pregnancy, especially when it comes to the end of pregnancy. The placenta produces another hormone that we'll talk about later, especially in the metabolic section. It's called HPL, or human placental lactogen, and it's an incredibly important hormone when it comes to ensuring that baby has enough energy, enough glucose, especially as it gets larger and its brain gets bigger towards the end of pregnancy. So we'll talk about that in sections to follow. Now, we looked at the development of that amniotic sac very early on, and the fact that it's filled with amniotic fluid. Now, amniotic fluid is an important component of fetal growth, and it's something that you might take for granted. But the fact that the baby is suspended in this fluid environment actually creates a perfect condition for symmetrical growth of the baby. It maintains a constant temperature and pressure, and it also provides an enormous amount of protection in terms of cushioning the fetus, absorbing forces and preventing damage to the baby that's suspended in the amniotic fluid. It also allows for the free movement of the fetus. It's in this sort of weightless environment where pressure is sort of evenly distributed, especially any pressure coming from uterine contractions. And the fluid contains antibacterial factors that really help protect the fetus from infection. And amniotic fluid is about 98-99% water, and the rest of the solutes in that water include things like electrolytes, creatinine, urea, bile, pigments, renin, glucose, hormones, and fetal cells. And so if we look at this slide, what we're actually seeing is that we've got two compartments. We've got the fetal compartment, and then we've got the amniotic cavity filled with amniotic fluid. And what we see is that the arrows that point from the fetus into the fluid represent sources of input, sources of amniotic fluid production. Okay? And the arrows that go from the amniotic fluid back into the fetal compartment represent outputs. So this really is depicting the circulation of turnover of amniotic fluid. So, and then some fluid actually can cross the amniotic membrane and be removed that way as well. So you see that we've got the lungs exchange amniotic fluid to a certain amount. You've got some loss of fluid through the head. You have some absorption of fluid through some of the membranes, some of the membrane barriers. But by far, the largest source of amniotic fluid is urine flow, so fetal urine. And the largest source of removal of amniotic fluid is through fetal swallowing. So that's very important because, there are conditions where the amount of amniotic fluid in the amniotic sac is either too little or too much. And so you can imagine that if the fetus has any sort of congenital anomaly in its renal system or in its urinary system, that the flow of fetal urine out would be impaired, and it might compromise amniotic volume. And you also might imagine that if there's any congenital abnormalities in the structure of the GI tract, that might interfere with fetal swallowing and the processing of amniotic fluid, the removal. So maybe in that case, amniotic fluid would actually accumulate. Okay. The other thing this tells you is that since amniotic fluid relies so heavily on urine output and fetal swallowing, that must mean that the ability to produce urine by the fetus and the ability to swallow amniotic fluid and pass it through the GI tract represents two systems that are functioning very early. Right? So these are two systems that develop very early, and at the very least, their capacities are there pretty early. And the last thing I'll say about this removal of amniotic fluid, or the take-up of fluid by the lungs and by swallowing and through the GI tract, amniotic fluid is also filled with hormones and growth factors. And so when these structures take amniotic fluid in, it actually stimulates the growth of those structures and the maturation of those structures. So it serves a lot of purposes in addition to just removing that fluid. So we know that there are different stages of embryonic and fetal growth. We know that we've got these pre-implantation stages and the very, very early embryonic stages. We know that we have post-implantation embryonic stages, including the organogenesis, the development of all the body structures. And then we also know that there's a fetal period, right, where these body structures are elaborating, growing in size, maturing in function. Now, there are, unfortunately, there are a multitude of birth defects that can occur. And these birth defects can be caused by genetic abnormalities and therefore an alteration in the development of the fetus. It can be caused by external factors, environmental factors, or internal factors. And when a birth defect is caused by basically by non-genetic causes, when it's caused by some internal factor like hyperglycemia is an internal factor, or an external factor like exposure to a drug or exposure to an environmental toxin, we call those substances teratogens. Right? So we have endogenous or internal teratogens and external teratogens. And the types of defects that can be caused by teratogens include things like malformations, where you have a physical alteration of structure. You can have deformations, where basically the shape or position of the body part is changed because of mechanical forces. You can have a disruption of the development of normal tissue. So a teratogen, the actual definition of a teratogen is really any substance, organism, physical agent, or deficiency state present during gestation that's capable of inducing abnormal postnatal structure or function. Right? So any substance, organism, physical agent, deficiency state present during gestation that induces abnormal postnatal structure or function. So there are many drugs that are proven to be teratogens. There are many environmental toxins that have been proven to be teratogens. There are many metabolic disorders that are proven to be teratogens. Right? So, and teratogens cause disruption by doing things like mutating genes, causing chromosomal breaks, by interfering with mitotic division or causing cell death, right? Or any number of mechanisms. It's not an exhaustive list. Now, in terms of whenever we're thinking about teratogens, we have to bring up vulnerable periods of time. So in this slide here, we've got the basically embryonic and fetal life broken up into three basic stages. There's pre-implantation here. There's main embryonic period, right, which is the 3 to 8 weeks. And then we've got fetal period, which is 9 to 38 weeks. So this graph is certainly not representing time in equal amounts, right? These columns are not representing time in equal amounts. You spend a lot more time in the fetal period than you do in these other periods. Now, the reason why this main embryonic period is drawn out to take up a lot more space is because this table is really trying to show periods of sensitivity to teratogens. And the most critical period of teratogen vulnerability is between week 3 and 8 of gestation. Okay. The pre-implantation phase is a relatively resistant phase. Usually damage or loss of the embryo at this stage is due to intrinsic abnormalities of the embryo, right? So chromosomal abnormalities or genetic problems in the embryo. Now this embryonic period, you can see, depending on the stage, right, different time periods, they reflect times where these structures are growing. So introduction of a teratogen during these periods of time can affect these structures, right? So for example, the development of the heart and all of the chambers and that very complex process happens between about 6 or 7 weeks to 8 weeks, right? So an introduction of a teratogen during that time will be more likely to cause heart problems than if it was introduced earlier or later. Okay. So you can see these vulnerable periods of time. And then during fetal development, while there are still some structures that are undergoing significant further development, like the central nervous system in very late pregnancy, external genitalia during the fetal development between 9 and 38 weeks, the palate, the teeth, the eyes. There are fewer structures that are undergoing critical development during this fetal period. Okay. So by far the most vulnerable period for teratogenesis is this embryonic period. Now, there are many, excuse the crooked nature of this, this table, but there are a variety of drugs that have been recognized as known teratogens, and they include a wide spectrum of drugs. Now that doesn't mean, so oftentimes the goal, if you need to use drugs in these categories, the goal is to find alternative drugs during pregnancy that serve the same purpose, if that's possible. There's a very sad story about a drug that was actually given widely to pregnant women and caused a severe birth defects is the story of thalidomide. So thalidomide was a drug that was sold during the late 1950s and 60s, and it was used as a sleeping aid for pregnant women and an antiemetic for pregnant women. So it was actually marketed to pregnant women to combat things like morning sickness and insomnia. Now, later in 1960 to 61, they found that thalidomide was causing significant birth defects in the form of this condition that they called phocomelia. And this is a birth defect, a pretty profound birth defect, where one or more of the children's limbs would fail to develop. So they would either not develop at all or they would be shrunken or deformed. So there was about 15,000 fetuses that were damaged by thalidomide. Um, and this happened throughout the world, not just in the United States. So about 46 countries had babies born with these birth defects. Okay? So thalidomide was banned, obviously, um, and it actually has found its way back on the market with, of course, very strong warnings against use during pregnancy. But I believe that it's being used as a sedative nowadays for things like leprosy and multiple myeloma.